Hi everybody, I hope you are all well and having a great week. Today we are going to discuss the importance of oocyte morphology and its effect on IVF outcome. As you are probably aware, selection of the oocyte with the best developmental potential has been the focus of intense research in the last few decades. The success of IVF treatment is dependent on the quality of gametes available for fertilization. The oocyte developmental competence plays a key role in appropriate embryo development. It is usually defined as the ability of an oocyte to mature and to be fertilized and sustain embryo development up to a live birth. These include some of the most critical and complex biological characters, such as integration of the male genome, nuclear reprogramming in the zygote, and activation of the embryonic genome. At present, comprehensive oocyte morphology scoring is not part of the IVF lab protocol. However, oocyte morphology assessment can be useful in embryo grading and selection for transfer and freezing. In addition, oocyte morphology assessment can be particularly useful in oocyte donation and oocyte freezing for fertility preservation cycles. Furthermore, in cases of very poor treatment outcomes, objective oocyte scoring can be very important. However, we know that not every morphological variation of oocyte may impact its developmental competence. On the other hand, even the most normal appearing oocyte can be masking aneuploidy. The abnormal oocyte morphological variations in cellular structure that may negatively impact their developmental competence are called dysmorphism. However, there are also morphological features that represent variation of normal oocyte morphology and have no effect on embryo development. How do we distinguish between oocytes that express morphological variation that have a negative impact on IVF treatment outcome and with those that have no impact? Firstly, I would like to separate the different features that can be detected in oocyte into three different areas: the cytoplasm, the perivitelline space, and the zona pellucida. Secondly, to divide them into those which have impact on IVF outcome and those who have no impact. In the cytoplasm, vacuoles of different numbers and diameters, dark color of the cytoplasm, homogeneous granularity, centrally located granular area, aggregates of smooth endoplasmic reticulum, and refractile bodies. In the perivitelline space, debris, enlargement, and fragmentation of the first polar body. In the zona pellucida, its irregular or non-spherical shape and dark color. In addition, the prominent types of abnormal morphology are giant and enlarged oocyte. Some morphological characteristics do not appear to influence IVF outcome. Oocyte with ovoid shape, dark-colored cytoplasm, or homogeneous granularity of the cytoplasm are not different from those that do not exhibit any visible morphological alterations and appear to have no influence on treatment outcome. Abnormalities such as refractile bodies, fragmented first polar body, dark zona pellucida, enlarged perivitelline space, and debris in it are likely to affect the treatment outcome to some extent. Finally, clusters of smooth endoplasmic reticulum in the cytoplasm, centrally located cytoplasmic granularity, and vacuoles negatively impact infertility treatment outcomes. Next, I must discuss the phenomenon of giant oocyte and its effect on embryo development. The incidence of giant oocyte is just 0.3%. However, they have a prominent negative impact on IVF outcome. There are two main known reasons for giant oocyte. The first one is a nuclear but not a cytoplasmic division of the oogonium. The second one is a cytoplasmic fusion of two oogonia, and therefore the result is tetraploid oocyte. Cytogenetic analysis has revealed that the unfertilized giant oocyte are diploid. This means that they contain double chromosome numbers. The zygotes were genetically abnormal even when they exhibited 2 pronuclei and 2 polar bodies. The question is, do giant zygotes have the capacity to develop to the blastocyst stage? It has been reported that giant zygotes develop to the blastocyst stage, even very perfect one regarding the morphological aspect. However, they demonstrated genetic abnormality. I would like to share with you a video of a giant oocyte developing into a giant blastocyst. You can compare its size to the normal one in the left well. As you can see, the giant oocyte contains about twice the cytoplasmic volume compared to the normal one, and it therefore covers most of the well's area. You can see the giant H blastocyst that goes outside the well's borders. Fertilized giant oocyte with 2PN should be treated like multipronuclear zygotes. In this case, the sperm nucleus is added into an existing diploid chromosomal content of the oocyte. Among chromosomal causes of human reproductive loss, triploid is the third most prevalent of all abnormal aborted fetuses. Embryos that develop from giant oocyte are at higher risk of the genic triploidy. Based on this evidence, giant oocyte should not be used for clinical practice. As soon as the oocyte size is considered, more cases of the genic triploidy can be identified and embryo transfers can be prevented. The essential question should be, what are the challenges and the future directions in the study of oocyte morphology? Currently, morphology-based microscopic methods continue to remain the main option for oocyte quality assessment. Embryologists rely basically on the maturity status of the oocyte or the presence of the first polar body. Today, oocyte morphological assessment is more informative than predictive. If we could integrate oocyte morphology data into AI algorithms, then the process of embryo selection would be much more accurate. This would prevent the transfer of abnormal embryos and increase the probability of implantation and live birth rates. Let's discuss this in our next podcast, as it is a very relevant topic. Stay tuned.